Yeah, so this has been a privilege for me to, I want to tell you, to work with Dr. Smale. And in fact, the title is Reprogramming. So in fact, last 2.5 years, I am reprogramming myself in terms of mathematics and in general. So, and I'm inspired by Dr. Smale. And it's good to have inspiration, it's always. Right, so as you see, the title of my talk is Algorithm for Cellular Reprogramming. First, let's start with this. So my goal. So I was a transform biologist, I will say. I trained as an electrical engineer, and then my graduate work was in mathematics. And last almost 10 years, I'm working on biology. So my real goal in my research is that I want to humanize my science. I want to humanize my science and really change the world. The idea is that, so my funding is coming from an institute called DARPA, Defense Advanced Research Agency Project in USA. Their motto is change the world. Their motto is that means that we can do the things that not trivial, to think like science fiction. So then first I will introduce one topic somewhat I am obsessed with last few years. So this is the topic. I don't know how many of you know. So far the best treatment is a high dose of chemotherapy. That means if you get a high dose of chemotherapy, you can kill cancer cells. But when you apply a high dose of chemotherapy, you cannot target it only for cancer cells. You also kill your immune cells. That means basically, if you get a high dose of chemotherapy, you wipe out your immune system. I don't know how many of you know that. Right. You have a chemotherapy for cancer, you wipe out your immune system, now you have another problem now. So that solve that problem, treatment is, it's called bone marrow transplant. So the bone marrow transplant is the treatment for the treatment. Right? You can think that way. Bone marrow transplant is the treatment for the treatment. So I was at Fred Hutchinson Cancer Research Center in Seattle for 6 years. That's the way our bone marrow transplant was developed. The person who got the Nobel Prize was there, Donald Thomas. So I will tell you a story here. Right? So what is bone marrow transplant? Let's say I had a leukemia. So I got a high dose of chemotherapy. Then I lose my immune system. Let's say somebody, Shuai, is my brother. So he will give me bone marrow cells to me to rebuild my immune system. But 80% of the time, his cells attack my body. That is called graft versus host disease, GVHD. That is the biggest complication in bone marrow transplant field. The donor cell will attack the patient's body. By the way, please disturb me anytime you like, okay? So that is a biggest problem in treatment for cancer. So then what is the ultimate solution will be? To minimize the GVHD. I don't need his cells. Can I use my own cells in somewhere, not cancer cells, some normal skin cells? Can I reprogram those cells and put it back as a bone marrow cells? So that's what I said. That is somewhat science fictional to many of you. Can I reprogram my skin cell to a hematopoietic stem cell and put it back? And then I don't need his cells. Everything is autologous, my own genome. The reason is that even my brother is not equivalent to me. I have my own genome, and all my cells have the same genome, right? Every cell, how I build, every cell has my mom and dad, right? Every cell. So the cells are the cells, genome is the same. But how is wired different depending on the cell type? So that's what I said, get a skin cell. Can I change that skin cell to become a bone marrow cell? Okay. So then, as I said, I really want to do that. I really want to do that. But people have done this too. I will tell you what people have done so far. And then I will tell you why mathematics is important here. Not just biology. It's an integration of beautiful biology to beautiful mathematics. Together we can solve this. Right. So let's go little deep into this. So who I am? So I came from the beginning. My mom and dad gave sperm and an egg, then create a one cell that give rise to all the possibilities in my body, all the cell types. Right? So that is the classical picture of cell biology. You have one stem cell that give rise to all the possibilities in my body. It's a forward direction. Right? If I may draw this picture in a different way, I think. Let's say you have a, say there's like a, you can think it's like a mountain, and you have a cell here, right here. This is the embryonic stem cell. Right? The first cell. And then this cell give rise to all the possible cell types in my body and your body. All the cell types that give coming from this cell. Right? That cell. So roughly, approximately 300, I will say, cell types in your body. Approximately. But maybe it's not true either. Yes?
So in your picture now, the yes cell is in like a minimum place, right? So you have to give it some simulation to let it change to other states.
Yeah, so that means naturally certain signals initially that create different paths to specialize.
Yeah. I mean, for this, I remember it called like Washington landscape.
Yeah, this is called Waddington's epigenetic landscape.
Yeah, so in their like assumption, do they have a local minimum, like a cell can choose our states and then it just stops?
Yes. So the idea is that somehow the nature, billions of years nature, they create that something called differentiation, cell to specialize. So that means the think about like this, how my analogy of thinking this is, let's say I am in high school, right? I have no idea what I want to be, right? So I am in most uncertainties in my, I have, right? But I have potential to be someone, right? So that is like right here, embryonic stem cell, right? I have potential, right? Then I go to college, I get a degree, right? I am more little bit specialized, right? And then now I'm a faculty, I am more specialized now, right? And more, there is a terminally, this is called terminally differentiated state. So that is the function, like kidney, liver, and heart. There are these 300 cell types really defines you, the function. Yes?
Does that final state indicate that they have like a minimal energy or entropy or something?
Sure, I will say that final state, if you can draw this axis in some sort of a, I will use the word entropy. Right? So more specialized, more become deterministic, I will say. More order. I will, as an example, to go to her question, your red blood cell. Your red blood cell has no nuclei, no nucleus. So that happened through something called HSC cell, specialized to a red blood cell, and nucleus ejected and become a red blood cell. And when it's ejecting, nucleus become a sphere. Right? So there's amazing dynamics. And to answer to your question again, this one has more potential, more uncertainty, and minimizes the uncertainty. So this is the classical picture. Right? It's called Waddington's epigenetic landscape. But in brilliant two biologists changed this paradigm. So one was one of my heroes. His name is Hal Weintraub. He was at Fred Hutchinson Cancer Center. What he showed in 1989, he showed that he can change this. He can specialize cell, he can switch to this. He changed the one cell type to another cell type. But he published his paper 1989 As you see, he passed away in 1995. But it was a very unexpected, and nobody believed that much what he done. Yes?
He changed the state directly from state A to B without going like,
Yes.
up.
Yes. Yeah. Directly. Going up. That's the whole point. From here to here. 1989. Then, 2001, George Bush banned embryonic stem cell research in USA. So there was a postdoc. He was Shinya Yamanaka was a Japanese scientist at UCSF in USA. So he knew his work, Hal Weintraub's work. Then he thought, why we cannot create an embryonic stem cell from skin cell? So that means he thought, to me, he was the most brilliant guy, Weintraub. But what he did, he pushed the system to here. Go back. So this is the way you might like the story. What he did, very simple. He looked at a signature for an embryonic stem cell from gene expression. You might know what is gene expression means, right? There are some techniques now you can see which genes are expressed in a certain cell type. So he looked at the embryonic stem cell and then he looked at the skin cell. Even though there are 20,000 genes in the body, certain set of genes expressed for embryonic stem cell, certain set of genes expressed for skin cell. So what he did, the most simple mathematical operation, just subtract, right? Just subtract. Like, let's say Shua is there, I am here. What is the difference between us? Right? Then as a biologist, he found 26 transcription factors. I will not go into that. Certain proteins make that difference, 26. Then what he did as a biologist, he tried to put one by one to skin cell and see what happened. So because he want to minimize that difference, right? And then magically after 6 years, he found there are 4 transcription factors, MYC, OCT4, SOX2 and KLF4. If you give these 4, you can change a skin cell to an embryonic stem-like cell. He showed that, and this is the brilliant part. He got the Nobel Prize in 2012. He got the Nobel Prize in 2000. It's a very simple thinking, right? Just minimize the distance. Yes, question?
I mean, if he discovered like a set of genes, right? So what he did is he tried like modify each gene at a time, like a cell, right?
No. Reprogramming is not modifying. Reprogramming means that in a skin cell, these 4 genes are not expressed. So you want to give exogenously to the cell, artificially. That's what reprogramming means. If you have a silence, you give artificially a protein to the cell. Right? And then he found 4, got the Nobel Prize in 2012. And Weintraub, same skin cell, he introduced a 1 transcription factor called MyoD. Then he changed to a skin cell to a skeletal muscle. So this is some sort of a paradigm of what people have done in reprogramming. So that gave us a recipe as well as the general thinking. We can change a cell type. We can change the cell type to something else. That is amazingly remarkable. But still, classical geneticists not believe this. Because the general paradigm is that one cell give rise to all, right? Can I manipulate that? So again, I will come to now later on mathematics. So this is some sort of area in I will say in electrical engineering called control theory. Can I steer a system the way I want? And I love control theory. I love dynamical systems. Like as example, let's say, can I steer you guys the way I want? I observe, let's say a social network. I usually always do that in my class. I study my students, right? And can I steer the way I want when I better understand them, right? So I developed this theme called network control in 2011, network control, right? So So I will come to that. But the general idea is that these guys gave us a recipe to reprogram the cell. So then, so our work, my lab, and our group work is from here, go to any cell you want. Can we maneuver cell types? Any cell. If you have a heart problem, can I create a heart cell from skin cell? As Dr. Smale pointed out last time, that in the heart, there are two cell types in the heart, cardiomyocytes and fibroblasts. If you have a heart problem, heart attack means you lose the cardiac myocytes. Can I use skin cells in the heart to reprogram to cardiac myocytes? Then you have a solution for that. So that means our goal is get a biopsy skin and change that cell type to anything you want. That is the science fiction, right? It's not science fiction really. You can do that. And more elaborately, I can show you this picture. Get a skin biopsy, any cell type you want. Can you go in? Because skin is already everywhere. Skin cell, my fibroblast skin cell. Right? But there's some sort of complication. I draw here neurons, right? The brain. Brain has many types of cell types, not a one neuron, right? But okay, sure. So I want to show you another. So what I'm saying is that last 4.5 years in my lab, we done some of this. So one of the main guy in my lab is Hemeng Chen. He's the first guy who reprogrammed a bipolar patient's skin cell to a neuron. There's a disease called bipolar disease. And you cannot study bipolar disease. You cannot get neurons from the brain. What Hemeng did, you get a bipolar, Hemeng and a group of us. Hemeng is the main guy that get a skin cell from a bipolar patient. He did the long way. Get a bipolar patient skin, make iPSC. So I want to write another word here. So the reprogramming, so as I said, fibroblast, right, FB, you introduce artificially this MIC, OCT4, SOX2, and KLF4. So these are called Yamanaka factors. Yamanaka factors. Then you can create this ESC, right? ES cells, but these are really called iPSC, induced pluripotent stem cell. You induce that state, right? iPSC. So what we did, create a bipolar patient skin, iPSC, IPS to neuron. So that was that experiment. But our real goal was, can we do intelligently, directly get me create a new, that is done now. People done, not us, people done. Directly converting a skin cell to a neuron, that's done. You can just Google and find out. Right? So again, go back again, right? So our goal is one of our topic. I really want to solve this problem. Create from skin to construct a hematopoietic stem cell, and then you don't have GVHD. There's no GVHD, right? So this is now one of our papers are published in last year called an algorithm for cellular reprogramming. Exactly the title of my talk. This was our team, very diverse group of people here, and we conducted this work. And I highly encourage you to read this paper in PNAS. And I really want, so now I will tell you how we are approaching this topic, reprogramming. I love technology. The technology means there are amazing way now you can interrogate the genome, not just gene expression. You can see inside the nucleus where the genes are. So this is really a quote I love, this quote, that any sufficiently advanced technology is indistinguishable from magic. So I will show you some magic, how to see inside, right? So for that I want to give you a small background about certain technology that developed in my lab as well as more general now. So one of the sort of technology is something, the concept of 4D nucleome, 4D. So the aspect of 4D nucleome is that, so as I said again, Yamanaka, what he did, he looked at the gene expression. Right? What is the output of a gene, roughly speaking, right? But in what we are interested in, how the inside the nucleus, how these 20,000 genes organize. So what is the geometry of genes, how they folded in 3 space. So this whole concept is we can image, there's a technique called chromosome conformation capture. It's a biochemical technique, you can map inside the nucleus what is the network underlying. Which genes is communicating with which in spatially, roughly speaking. So this whole concept, we invented this term, 4D nucleome. Thomas Reed at NCI, he will be a speaker next week, and myself, to study genome as a dynamical system from data. So that is some sort of our approach for reprogramming. I will come to that. And also, I think Dr. Smale inspired me all the time that I'm a more thinking deterministically, how things are inside my cells. And one of the examples I want to show you here, so the cell cycle, I don't know how many of you know that. Cell cycle is how one cell creates 2 cells. In naturally, my body, every second, millions of cells die. Millions of cells die. Your body or my body, millions of cells die. But new cells come back and keep still me, right? Still, new cells will born and still me. That process is called cell cycle. This cell cycle is so important for reprogramming, to understand the cell cycle, right? So cell cycle means this is called G1, different phases. So I think majority of you know chromosomes. What is a chromosome mean, right? Chromosome. Right? You know chromosome. But I think majority of you know this picture of a chromosome. So you know, in the textbook, the chromosome is like this, right? Like this. Right? This is the picture in the textbook picture of a chromosome. But that is only happening in one stage of the cell cycle. That is only happened during mitosis, M phase. During these other phases called G1, S and G2 called interphase, chromosomes is not like this compacted form. It is a cloud in 3-dimensional space. It's called chromosome territory. The chromosome is in a cloud, like a beautiful cloud. Right? There are 46 clouds floating around inside the nucleus. So that is so important to understand that. It's a beautiful, so cells have these clouds, they communicate each other, then clouds go through the cell cycle and compactified like this and divide. Right? I really encourage you to think and read about the cell cycle since you are interested in genomics. So important aspect is cell cycle in natural in genomics. Okay, you with me here, right? So this is to show that. In fact, there's one more thing, little bit unrelated to my topic, but I want to tell you this. There are 2 chromosomes in, say, chromosome 1, you have one from mother, one from father, right? They are not even close in space. This is an image of, say, this green is chromosome 7, they are far away inside the nucleus. There are a lot of mysteries inside the nucleus. Okay? And let's go to this. I want to briefly, again, I need this technology for reprogramming. This chromosome conformation capture, biochemically. So the biochemically you can map, so let's say this is a 1 cell, okay? 1 nuclei. So if you zoom a small portion, so let's imagine this class is the nucleus, right? We are genes, right? I want to know in a given time who is close to who, right? So what the technique is, I give a glue to you, and then if he and I are close, there's a glue bond happen, right? This is like this bond, right? This is close to this, right? Then using sequencing, I can construct a map for the entire genome, who is close to who. Right? So that is this map. So that is interchromosomal contacts with us intrachromosomal contacts, both ways. Because I told you before, if you recall that, we use the concept of network control. So this data will naturally give me some aspect of a network. Right? It's a contact map. It's give me a network. It's not gene expression. Inside, I have an idea who is close to who, right? You with me? So also that data also tell us there's an amazing organization principle inside the nucleus. Amazingly organized. I have to use certain words, but I will not explain. You can just Google and find. There's a term called transcription factories. Have you ever heard that word? Transcription factory. Maybe not. Please Google and check it out. If you want more reference, send me an email. I will send it to you. So important. The idea is, there is something called nucleolus. Anybody have heard the nucleolus? Maybe not. The nucleolus is the something called that nucleolus make ribosomal RNA. So the nucleolus is made out of something called nuclear organized regions, NOR. They are spread between 3 different chromosomes in human, but they come together during the cell cycle to produce a factory. And likewise, now people tend to believe there's a sub-factories inside the nucleus. Those are called transcription factories. The chromosomes go to those factories dynamically and use those machinery to express. So that means naturally it's organized inside the nucleus. And again, please, so this is the, his name is, no. You check it out, his work, Peter Cook at Oxford. So he coined the term transcription factories. Right? So the idea why we need that concept, this is the high C matrix I show you, high C. But if you see the high C, this is a product of 1 chromosome. Okay, 1 chromosome. You can see there is some sort of a block structure to high C data. Some sort of block structure. Right? What does that mean? That means there are some clusters, right? Some clusters. Diagonal, block diagonal, right? So that means if you think more like a mathematically, what you can conclude from this picture? What you can conclude this picture? That means some sort of a dimension reduction, right? You can think as a dimension reduction. There's a certain way they have certain relationships that means I can group one part here, one part here, one part here, right? If you can group, then I can get some sort of mean of this to represent that group, right? So the roughly speaking, there are in the human genome, that boils down to roughly 2,500 block structures, roughly. 2,500. These are called topologically associated domains, tags. We have another paper on this topic if you're interested in 2016. So now, okay, little bit you with me. So why I need these things? Because I need to tell you, so our goal is to reprogram, right? But we need to understand first what is the organization principle inside the nucleus. Then I have a good idea to intervene to that. Not like what Yamanaka did. He was brilliant to show us the reprogramming is possible. But we want to do more intelligently. Right? So this one aspect is to understand organization principle inside. As I said again, if I want to study some social network to intervene, I want to study how they related to each other first. Right? And then I can intervene to that. Maybe it's not a good thing, but what I'm saying is like that's the way it is, right? If I want to, control theory means exactly that. Right? So also we also so in my lab in 2014, what we did, we thought we really want to understand how the skin cell go through the cell cycle. How a natural skin cell go through the cell cycle. What is the dynamics? So what we did, we got human skin cells. We synchronized the human skin cells to G1, and then we allow those cells to enter to the cell cycle. And every 8 hours we got samples for 48, 56 hours. And we measure that technique of chromosome conformation capture and normal RNA-seq. So what we observed was amazing. So that is some sort of 4D point of view of the cell cycle. 4D, time, structure, function, right? So what we observe, if I really summarize this picture here, so these are this block structure. So this is a one block structure. Okay? The TAD I call that organized. So just let me see. So this is a one block structure. This block, these three are three genes in that block. You with me? In that one block I show you, the genes are inside. If you look at those genes with time, their expression, they tends to synchronize the activity. In this block, there is all a silence. So that means that the block structure is a reflection of the underlying expression pattern. So, but some blocks, of course, you don't see that. Some blocks, of course, you don't see that. So we use this was beautiful finding to us. That means the genes belongs to those blocks tends to synchronize the activities naturally. And also more detail in this paper, 2015, PNAS, it's called 4D nucleome of human 4D nucleome. Oh, I will not go into that. But I want to touch this somewhat because I said the cell cycle, right? Cell cycle is so important for reprogramming as well as anything you do in biology. That's how I think. Because that's naturally one cell. As I said, I want to tell you again. Every second, millions of cells die. New cell will come back, right? So that happened. Amazing control, amazingly controlled process. You can say this picture I will make. I have a lot of time, so let I spend some time for this too. Right? So here, let's imagine this is at G1. I show you the cell cycle, right? Let me draw again. Let me draw again. This. Right? So my goal is to tell you. One of my goals is to excite you with biology and math. That's my goal. One of my goals. And also I want to show you the technology is amazing. We can do anything is possible. Yes?
A side question. So if we synchronize all those cells at G1 phase, will they enter the G2 or S phase at the same time?
Yes.
You sure?
We can measure from experiments how much went into the G1, but that synchrony loses with time because we are artificially creating this synchrony population of cells.
Yeah, but you only can make them start from the same point, right? You can't guarantee they go with the
No.
same speed to that.
Have you done any experiments, may I ask?
Okay.
Okay.
Yeah.
Please read our paper. Okay. Yeah. So if I may draw here M, G naught, G1.
Color.
Black.
You can see this? This is the cycle in G naught, G1. This is S phase and this is G2. So let's see, I want to draw this. So here, right here you have a one cell, your cell, one cell. You have one genome here, one genome, one cell and a one genome. During this S phase, there's a birth of a new genome happening. So at the end of S, you have a one cell, there are two genomes operating there, inside the nucleus. There are two genomes. Even G2, one nuclei, two genomes operating. Right? So if I draw this one here, right here, right? So this is a one genome. There are two genomes at the end of G2, and when cell divide, you separate two cells. Each has their own genome. So this happened there are many possibilities this can happen. What I meant by that here, so let's say you start with a capital A cell that can divide into AA, right? Purely symmetric. One cell give rise to same cell, right? You with me, right? And also that you can divide into A, B. That's an asymmetric division, right? This is good, right? But also you can divide into BC. And also maybe you can divide into BB. Right? There are four possibilities, right? To me, this is I want to achieve in reprogramming. Get a one cell, I want to make them to divide two cells, they are different. Right? This is reprogramming. This is my goal. This one. That's what I said. This is reprogramming. And also I said this is so important. Why? Sometimes this can happen, right? So this is like cancer. You lose the predictability of cell, how they divide. And this is purely, I really like this quote from Hartwell. Cancer is a disease of accuracy. If you lose the accuracy of the cell cycle, cells start to go wrong. Right? And I will make a very broad statement then. Someday somebody will reprogram a cancer cell. Right? We can. We can acquire control over cancer cell. Absolutely. Very, very, very soon somebody will do that. In fact, people are doing that, thinking about that. Okay, so this is this picture, please internalize. I'm just saying. Right. So now let's go back to, so you know some ideas now, right? What we use, chromosome conformation capture, Hi-C, that Hi-C technology, and something about cell cycle. And now this is the mathematical way of looking at reprogramming. So this is very, very simple, very, very simple mathematics. Right? So this is, you have some sort of initial configuration, you have a desired configuration. What I meant by that, your desired is say a cardiac cell, my initial condition is a skin cell. I want to steer this to here. So this is very simple linear system. This is all linear. Right? This is well studied. So without this part, so I will redraw again this picture again little bit differently. So let's imagine, I told you my set of genes in any cell is the same set of genes. Right? So imagine my genome has 3 genes. Right? Let's say this is fibroblast. There are 3 genes, and let's say their network underlying is like this. 3 genes. This is the network for fibroblast. Let's say this is a cardiac cell. Right? Let's say my network is now. Let's be precise here. This is gene 1, 2, 3. 1, 2, 3. This network is like this. They are different, right? How I can change this configuration to this configuration? So what I want to put into to do that, right? And which node I want to do that? That's the key. Which node I want to do something. So that part coming from this. Naturally in my body, as I said, 300 cell types, I will say. Maybe 1,600 to 2,000 transcription factors, right? So very finite number of transcription factors and finite number of cell types. That means we can do a lot of things. And the idea here, again, transcription factors, these are called master regulators. Subset of transcription factors can completely maneuver the cell. So I have a paper with Dr. Smale too, if you are more interested in mathematical side, called Mathematics of the Genome. Please read that paper too, Mathematics of the Genome. We lay down the idea of hardwired genome, how the genome is hardwired, how we can steer. But I'm not going to that one. Please read that one. Mathematics of the Genome, that paper, right? So the basic idea very simple, right? How I can intervene. Do something, rewire this to this, right? So this is, you can, now the thing is that we need to use data. We need to use data to do that. I told you we have skin cell, right? Proliferating fibroblast in a discrete times, right? So our idea was to think even more discretely. Each time point I study the dynamics, right? And if I write this more genome perspective, our goal is given any desired cell type which transcription factors need for conversion. That's the key, right? Classical problem, you have initial condition, desired, difficulty, what is X, what is F, what is U? Those are the questions. What is X in that equation, right? And we have data. RNA-seq, high-seq, transcription factor binding. And also there are a lot of databases. Binding, in fact, yesterday Dr. Baldi mentioned that he has a big database about transcription factors. Which transcription factors recognize the one-dimensional genome is some sort of cell type invariant. But if you superimpose something called chromatin accessibility, I'm not going to that, then you can find out which transcription factors influence which segments in the genome. So these are the things we knew about our algorithm. We required that. And I will give a very simple example, a little bit motivate you. Right? Naturally, what Weintraub, remember the Hal Weintraub, what he did, naturally means that picture I draw, Waddington picture, in something called myoblast. Some sort of myoblast, a cell type, that become something called myocyte, and myocyte become a myotube. This is equivalent to a skeletal muscle. So that is the nature, always do that. This is nature. This is the nature. That is called differentiation, specialization. In fact, this is coming from something called satellite cell. Satellite cell become a myoblast, become a myocyte, become a myotube. That's the nature does that. But Weintraub did, he studied the nature, how this do that nature. Then he found in this point, myoblast state, there's a one transcription factor completely acquired control over this process. He knew that MyoD was that master regulator, MyoD. That completely acquired control over myoblast to become a skeletal muscle. So then he knew, he went to a skin cell, a different lineage, and he looked at whether the MyoD expressed there. MyoD not expressed. So then what he did, he put MyoD artificially, exogenously. So that is the reprogramming means, right? The MyoD, that protein is, there are no protein, that gene is silenced, you artificially give. And then this MyoD, like master regulators, can regulate themselves. That's the key. Some, like us too, right? Some we can excite ourselves sometimes too. Like master regulators, they can regulate themselves. So what happened was when we introduced MyoD, then boom. Yeah, you have a question?
Yeah. Like in most of the cases, you can find a bunch of transcription factors that are correlated with each other, but how can you find the master factors? How can you identify which is like a master?
Good question. We don't know how many master regulators exist still in the human genome. We know MyoD is one of the master regulators. MYC is a master regulator. Some GATA1. There are finite number of master regulators we know. We don't know all the master regulators in the genome.
Yeah, I mean, yeah, you can identify them from like a pathway, like they are in the upstream or something. But how can you identify? Maybe you can identify mathematically. Like?
Not mathematically. That's what I said. Those are the ones regulate themselves. Not all the transcription factors has binding sites near to their promoter. Not all the transcription factors. Some transcription factors has the motifs near to 5 prime. Not all the transcription factors. So those are maybe the master regulators. So this is the as I said again, reprogramming is we more and more understanding how the nature does that, and then we are engineered that artificially. That's the reprogramming means here. Maybe this is some cases it will be bad someday, but for us we want to use for good purposes, like minimize DVHD. Maybe I will give you another examples later. Those are the cases we are interested in. Not to build like a super somebody, changing someone, right? So again, so I'm going a little bit detailed, but you can find from our paper, but our again, so what Yamanaka did, he looked at these 20,000 genes to separate out the fibroblast and embryonic stem cell. But we are approaching more low-dimensional picture in a TED space. Remember the word I used called topological associated domains, that there's an organization structure inside the nucleus. So our comparison we do in that scale, because that is somewhat manageable mathematically. 2,300. That means the nature, to me, nature builds certain way to low-dimensional picture in a high-dimensional 20,000 space. So this is how we use this, and then our algorithm is that we use the cell cycle. As I said that what Yamanaka, again, go back to Yamanaka. He look at the skin cell, he look at the embryonic stem cell. Just 2 points. But we did, our algorithm, we knew how the skin cell go through the cell cycle, different stages. We had the dynamics of fibroblasts. We used those information for our algorithm. Cell cycle. And next picture is again using another. There's a more active area in my lab is to really to construct from data difference between cell types. How to compute differences between cell types. So in our published paper we had only 56 cell types. But recently we have now almost, like I will say, 200 cell types, distances between cell types. So our goal is how to, we know the difference, then how we can minimize the difference. And if I present to you more single slide version of my talk is right here. Single slide talk. This is all the things I told you is right here. Right. So we use this aspect of high C, gene expression tags, and then we define a new state variable called this functional unit, roughly 2000 functional units. And then we use a lot of publicly available databases. And then we were able to, just to show that the Yamanaka as well as Weintraub was right in our algorithm too. They came up. Right. So MyoD, and now recently if you recall that Yamanaka factors was not nano, OCT4, SOX2, KLFO and MIC. That was 2007. But 2014 people found only need 3. Our algorithm also predicted that, just 3. And again, I just would not want to show you that. So that's some I hope I convince you, you can reprogram a SAT, right? You can reprogram the SAT. So the goal of my lab and our vision is, one of our goal is to minimize GVHD. The other applications currently is, we are starting to do this project now, reprogram human skin cell to a B cell. That's the immune cell. The other, reprogram cancer stem cells. And this is a really hot area in cancer. There are a subset of cells in cancer called cancer stem cells. They are hard to treat. That means if you have a high dose of chemotherapy, only kill proliferating cells. But this is a whole area. Cancer stem cell means the small number of cells in cancer, they are in like a dormant state. So they can come back and repopulate again the cancer spectrum. So the idea is, can we reprogram those cells, cancer stem cells? That's the active area. The other one is this reprogramming human fibroblasts into liver cells. So that we done in the lab recently. We were able to create hepatocytes from skin cells to screen drugs. I will not go into that either. So what I'm trying to say is that this is not just, I am not showing you just any mathematics or data, but we can do this. We can reprogram cells. And maybe I don't need that. But finally, I want to come to this one, that Dr. Snail and I have been working on a topic called emergence of functions, right? So the real idea is not just to reprogram one cell to another cell type. We want to create an organ from one skin cell. Can I create a whole tissue? So that's the way we have one paper, and we are thinking another we are working on, not just genome, how the cell-cell communication. That means one skin cell, can I create a whole tissue? Not only can I create an organ, artificial organ. And if I go more broad, I told you I love science fiction. Can I print something at the end? Get a skin cell, I will print you the organ. Science fiction, right? So I think it's okay to think, like dream high. I always dream high. And again, please, you can. So I am here till next Tuesday. If you have more questions, we can discuss. And thank you so much for listening.
Questions?
Michael?
Yeah, thank you for the talk. Could you say a little bit more about how you find the matrix speed? Because you have initial cell, and target cell, and you illustrate how you get A and the B matrix. Because
Yes.
you
Yes.
control path.
Yeah.
And
How to get the B matrix.
How to get
Yes.
a
Yes.
little bit more.
Yes. Yes. Michael. So B matrix is the one, the transcription factors, these transcription factors, in a one-dimensional genome, this is the one-dimensional genome, they can come and do something. They can bind. This picture is cell type invariant. Which transcription factors recognize the one-dimensional genome? That is, we can construct from publicly available data, and there's another technique called ChIP-seq. There's a chemical technique which places a certain transcription factor bind. So B matrix basically is a, so these are columns are transcription factors, and these places is the 2000 my tag CAD domain. But we want to know in skin cell, so at the end of the day, inside the nucleus is very, very simple. It's like a telephone wire. You have a compacted or loose. The loose one is called euchromatin and a compacted form is called heterochromatin. Only euchromatin regions can be impacted by a transcription factor. So that information we can extract it from high C, which places are accessible to do something. So then these transcription factors, we create vectors which places they can intervene. That's how we construct the B matrix. So
and then how you determine the you have different combinations.
yes.
Right? You actually can have some little some are more optimal than others.
Yes, so not just the one transcription factor. This is a group of transcription factors. As well as, Michael, not just, so we need to introduce this, that's why I will go back to this. So, as example here, so I didn't even talk about the D matrix here. Right? So we want to, that means our algorithm, sometimes we introduce a one transcription factor at here, the next one at here, and the second one at here. Different stages too. Like Yamanaka did, he flooded with 4 factors. Because chromatin is accessible different from stage to stage. Right? So I can tell you more about the construction of D matrix, but again, our goal is that the current existing techniques, the efficiency of reprogramming is really low. That means if you start with 100 cells, roughly 20% or less than 20% reprogram. Our goal is, I will say, for me, I want to make, as I said, give me 1 cell, I will give you 2 cells, they are different. We use the cell cycle. Right? 2 cells are different. Thank you.
Yeah,
Yes, sure. Yes.
so we want to find transcript factors to reprogram. So I say you want to increase the expression. Is there any case that you want to decrease the expression?
Not the expression, but efficiency.
Yeah.
Decrease the
product from the you want to increase the expression.
No. So what I meant by that, usually, in fact, the Yamanaka's initial experiments, if you start with 100 fibroblasts,
Yes.
only 5% of the cells become induced pluripotent stem cell.
Okay. Right.
And Hal Weintraub, conversely, the direct reprogramming,
Yeah.
fibroblasts starting with 100 cells, 60 to 70% of the cells reprogram.
Okay.
What I said that we want 100%, not 100%. Give me 1 cell, 2 cells are reprogrammed.
Okay. Right.
Because we can only get a tiny biopsy from the skin. Yes?
Thank you so much. I just wanted to know whether in the cell cycle process, when you have 2 genomes within the same cell,
Yes.
are both of them being actively transcribed?
Very, very good question. I don't think we know that. Some genes are, in fact, some genes in the genome are biallelic, some are monoallelic. Right? So that means even G1, only one genome will express. Right? Those are beautiful mysteries. We don't know, we really don't know the cell cycle, how it's operating. I tend to think that even G2, all 4 copies will express in certain cases. If you send me an email, I will send you some good papers on this topic. You have my email number, I can write here. If any topic you are interested, please send me an email, okay? At r@umich. Please send me an email. Any of these topics, I will send you papers or anything you like. And the slide deck also, I can give it to you. I just want a final comment. This is so exciting area to be in. The interface between our theme is genome, cells, and mathematics. There's so much to be done, right? I wish I can go back to graduate school and to do. But you guys have an amazing opportunity to do change the world. Yes.
So, how many time steps when you model this cell cycle for the dynamical system?
So that one, 8 time points.
8 time points.
Yeah, but now currently our lab, we are creating every 4 hours. We are collecting samples now every 4 hours. So the roughly cell cycle is Yuan cell cycle is roughly 24 hours, roughly. Right?
You see, right?
So, but now we are almost done, like every 4 hours time we are collecting samples now.
So when you use the TAD to do the dimensionality reduction,
Yes.
you make the group of the
Yes.
gene expression, use the mean gene expression
Yes.
within the group.
Yes.
So that's equivalent to say you assume the A within the group will be a constant. All the gene
Yes.
within the group share
Yes.
the same A.
Yes.
Do you think maybe because if your block has certain diagonal, hierarchical diagonal
Yeah,
structure,
you are asking a really good one. So now we are, since then, we have more extensions. Now we are talking about TAD interactions too.
I see.
In fact, that can be interchromosomal as well as interchromosomal.
I see.
There are a lot of expansions since that our paper we published.
Yeah, is sparsity important for A? So here you did not assume any sparsity. Maybe some some element you can make it 0, indicate some interaction may not be that significant. That can be some regularization.
Yeah, maybe I would love to do something with you on this area.
Okay.
From the data.
Okay. I'm just curious. I'm not an expert. Maybe some.
Yeah. But there's a beautiful structure to the genome. That's given. It's amazingly organized inside the nucleus.
So you can verify the structure of A from some knowledge, biological knowledge.
Absolutely. There's another technique we are exploring. There's a technique called imaging. And I didn't show you these things. There are ways we can image the active area and the inactive area inside the nucleus.
I see.
In fact, we are starting lively image these things. I have a new microscope to live cell imaging
I see.
during the cell cycle.
Okay, so I guess we have heat now, and that's again.